Hello everyone, students. Good evening, all of you students. Welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher students. Quickly, let me know in the chat right now if I'm perfectly audible and visible to each and every one of you students. Quickly, let me know in the chat right now, all of you. Good evening. Hello, Bot Army students. Hello, Bot Army. How is everyone doing? And students, let me tell you honestly what happens. All of you love the chapter of human reproduction. Yes or no? You're like, oh, human reproduction. I can't wait to study human reproduction. Okay. Starting it is very easy. Primary sex organs, secondary sex organs. We learn about the entire male reproductive organ, the female reproductive organ. But as soon as we come to the gametogenesis, many students hate the chapter because gametogenesis is a process which many students try to by heart, many students try to mug it up, and doesn't work. It will not work, students. It will not work. Okay, hello Kavya, hello all of you, right? So if you try to by heart this chapter, never motte, never you will understand, students. So if you really want to understand this chapter, if you really want to master the spermatogenesis and the oogenesis, quickly, students, add fire emoji in the chat, all of you. Students, quickly add fire emoji in the chat because, students, let me just show you one more time. Let me just show you. Try solving these questions. Let me just give a demo. Students, tell me the answer. Can anyone tell me the answer, students? Can anyone tell me the answer here? Hello, hi Darshini, hello Ramesh, students, all of you. Array, DNA batch students are also here, students. DNA batch students are also here today. Okay, students, try solving this question. Difficult, no, difficult, yes or no? Okay, try solving this question. Okay, try solving this question. Tell me, like, such questions will eat your head. It will eat your brain cells. Yes or no? Yes or no, students? So if you want to solve all of these such questions, these are quality questions. If you want to solve these questions, you need to watch the lecture till the very end, students. Till the very end. So, students, shall we start? Students, shall we start, all of you? Shall we start, all of you? Shall we start? Yes, students. Can I get a green heart in the chat because of the bot army, students? Bot army love. Can I get a green heart in the chat for the bot army? All of you. One students, I'm only confused. Okay, I'm only a little confusion. Life is only confusion, students. Okay, and students, quickly smash the like button, all of you. Not once. Oh my God! Like notification. Friends, please smash the like button. I want everyone to keep a reminder. Okay. Now, students, the first of all, understand gametogenesis has two parts: spermatogenesis and oogenesis. So I want all of you to know this spermatogenesis. It happens in the case of male, while oogenesis happens in the case of female. Male and female. Now, what is the need, students? What is the need? Like, what is the need of entire this process? The main need of spermatogenesis and oogenesis is to form gametes. Do you remember in the first class I told you a primary sex organ should be able to produce hormone, should be able to produce gamete. So this is how it forms gamete here. Okay, this is how it forms gamete, students. So the first thing is called as spermatogenesis. Spermatogenesis is a process where the sperms are produced. Yes or no? Okay. Now, students, let me just ask you one simple question. In the seminiferous tubule, inside the seminiferous tubule, right? Seminiferous tubule, there were two cells. No, who can tell me? Lakshmi, Sundaran, yes, Nandu, Darshini, Punya, Vibs, right? Who can tell me? Inside the seminiferous tubule, the lining, we had two type of cells. One is called a Sertoli cell, nurse cell. Function, it forms, it provides nutrition to the male germ cell. Okay. Now, which is the second cell, students? The second cell there was the male germ cell. Male germ cell. Now, can anyone tell me what is the other name of male germ cell, students? What is the other name of male germ cell? All of you. Okay, that is where the spermatogenesis starts from. Spermatogenesis starts from the male germ cell. Students, it starts from the male germ cell, which is also called as spermatogonia. spermatogonia. Now students, in examination they can also ask you what is the ploidy. Who can tell me? Huh? Any smart people in the chat? Not Leydig cell. Nandu, Leydig cell is on the outside. Leydig cell is on the outside. Remember, Leydig cell is called as interstitial cell. It is on the outside of the seminiferous tubule. Okay, produce androgen. Now students, remember the ploidy of which color? Okay, the ploidy of spermatogonia is diploid in nature. By chance, if they ask you, you should know. Okay, and students, this entire process of your spermatogenesis, it will start at puberty. Whenever there is an increase in GnRH, whenever there is an increase in not posting 46, I thought 4n. Okay, whenever there is an increase in GnRH, it happens. Okay, now students, now this particular spermatogonia, okay, initially it undergoes a process called as mitosis. Mitosis. Now students, tell me in the chat quickly, in mitosis, is there a change? Pink color, I'll use pink color. I will use pink color, don't worry. Okay, initially, students, tell me, in mitosis, is there a change in ploidy? No. Mitosis is called as equational division. So this one cell will divide into two cells, but it will remain diploid. It will remain diploid as such, students. It will remain diploid as such, diploid cells. Kutti diploid cells, students. Kutti diploid cells. Now, students, one such cell here, one such cell will become huge. It'll become huge. Okay, it'll become huge like this, huge. That is, one such cell will show growth. It'll show growth here, proliferation. Okay, it become bigger in size, bigger in size. Now, this cell is called as, friends, pink color I'm using. It is called as primary spermatocyte. Primary spermatocyte, students. Primary spermatocyte. Now, students, can anyone tell me in examination if they ask you what is the ploidy of primary spermatocyte, students? What is the ploidy? The ploidy of primary spermatocyte is diploid in nature. All of you know right now. One PYQ done here only, students. One PYQ done here only. Okay. So let me just write it on this side here because I need space other side. Okay. We get primary spermatocyte. Now, students, this primary spermatocyte now undergoes a process called as meiosis. Ooh, meiosis. Scary, no? Scary. Don't worry, students, I'll teach you meiosis. Now, students, you tell me in the chat right now. Meiosis is called as what? Reduction division. Will there be a change in ploidy? Yes, there will be a change in ploidy. So, this one cell, when it will divide into two cells right now, two cells, will they be haploid or will they be diploid, students? Okay. Yes, more than one cell can undergo mitosis. Again, Parky, doesn't matter. There are many cells which undergoes mitosis. Millions of sperms are produced. Remember, millions of sperms are produced. So there are many cells which are undergoing meiosis. Okay. Now, students, we get two haploid cells, meiosis. And once it undergoes meiosis, these two haploid cells are called as what, students? They are called as secondary spermatocyte. So from primary spermatocyte, meiosis, secondary spermatocyte. Now let's say, students, this is your 22 plus X chromosome, 22 plus Y chromosome is there in this, just for reference. Okay, Paki, okay, Paki. I told by mistake something else I told you. Students, in examination, if they ask you, boss, tell me, what is the ploidy? Yes, diploid means 23, haploid means diploid means 46, haploid means 23. Okay. Now, if someone asks you in examination, tell me right now, what is the ploidy of secondary spermatocyte? Tell me your answer, students. Tell me your answer. All of you in the chat, very active. Then, students, tell me the chat quickly. If someone asks you what is the ploidy of secondary spermatocyte, what is your answer? Secondary spermatocyte, Basavaraj Sir told us. Basavaraj Sir told us it is going to be haploid in nature. It is going to be haploid in nature, students. Okay. Now, students, now we will draw this side. We will go to cross country border. We will make one border here. Okay. Now, students, now I will take this secondary spermatocyte. Secondary spermatocyte, students. Secondary spermatocyte. Now, students, this secondary spermatocyte right now. Bro, I need more space, students. I need more space. Students, do all of you remember? Do we have meiosis 1 or 2? Do we have meiosis 2? Yes or no? Yes, we do. So this secondary spermatocyte undergoes up right now meiosis 2. It undergoes meiosis 2. So this becomes meiosis 1 right now. Meiosis 1. Meiosis 1. Now students, tell me in the chat right now, will this become n by 2 right now? Is it possible it becomes n by 2? Never. This cannot become n by 2 right now. No. Now this actually again give rise to 2 cells only. This will give rise to 2 cells. This will give rise to 2 cells. 2 cells here. Okay. So we end up getting, students, we end up getting 4 cells here. This will divide into 2 cells. This will divide into 2 cells. All of them are haploid in nature. And students, do they look like sperm, students? From any angle, you see one top angle, bottom angle, corner angle you see. Tell me, do they look like sperm right now? They do not. Where is the head? Where is the tail? It is not there. So students, these structures are called as spermatids. Spermatids. So students, if they ask you in examination, spermatids are a result of mitosis, meiosis 1, meiosis 2, none of the above. What is your answer? All of you. All of you tell me the answer, students. Tell me quickly in the chat. If they ask you the question, spermatid is the result of mitosis, meiosis 1, meiosis 2, none of the above. Meiosis 2. So spermatids are produced as a result of meiosis 2. Now students, now we will do one makeup now. Touch up, we'll do touch up. Okay. Now there is a process called as spermiogenesis. Students, do all of you remember? Remember I was teaching you when I taught you about Sertoli cell helps in something? Yes. Sertoli cell helps in something? Yes, it does help in something. So here we have spermiogenesis. Spermiogenesis. Now after the touch up and makeup here, they will start to look like sperm now, students. Now these are called as sperm or spermatozoa, same meaning. So we end up getting haploid sperm or spermatozoa. Now a question arises, does the sperm is released as such, like take the sperm, throw it out? Not possible. Now all the sperm which are produced, right, they are produced inside seminiferous tubule. All of these sperms and this spermatogonia, it is present inside seminiferous tubule. Now the head, students, the head of the sperm is actually embedded. It is embedded inside the Sertoli cell. All of you remember, students? All of you remember Sertoli cell? All of you remember, right? Yes. Do all of you remember Sertoli cell? The head of the sperm, students, the head of the sperm is embedded in the Sertoli cell. Let me just show you, students, here. Can you see this diagram, students? This diagram here. Can you see, students, here? This is the head of the sperm. Can you see it is present inside the Sertoli? This is called a Sertoli cell. This is called a Sertoli cell. Now the entire head is inside Sertoli cell, like this. Take the head inside, like it is inside the Sertoli cell here. Okay, the head is present inside the Sertoli cell. Okay, let me just draw Sertoli cell here. This is Sertoli cell here. The head is in Sertoli cell right now. Head is in Sertoli cell. Now students, now listen to me very carefully. Last point here. Okay, last point, it is over. Last point. Okay. Now students, now release of sperm from seminiferous tubule. Release of sperm from seminiferous tubule. Students, is it visible to all of you? Yes, it is visible, students. It is visible to all of you. Okay. Now, release of sperm from seminiferous tubule. That entire process is called as what, students? I'm using pink, be happy. It is called as spermiation. Spermiation. It is called as spermiation. So I want all of you to remember the process of your, what is spermiation, what is spermatogenesis, spermiogenesis, and spermatogenesis. Three different things here: spermiation, spermiogenesis, and spermatogenesis, and that is the end of it. Be happy, go home, good night. Students, that is the end of it. Students, how many of you understood? How many of you understood? Can I get a quick fire emoji, students? Can I get a quick fire emoji in the chat if you understood the entire process? If you want to learn spermiogenesis, VBITS, I'll teach you separately, because that is an entire big process. It is a very complex process, right? Students, what is this weird white light coming here? Students, I'm seeing it for the first time now. One weird white light is coming here. Stupid white light. Okay. Students, clear, all of you? Okay, define all. Okay, let me just define all of them. Let me just define all of them. Define. Spermatogenesis. Spermatogenesis is a process how sperms are produced. Sperms are produced from male germ cell. This entire thing is called as spermatogenesis. Okay, done. What is spermiogenesis? Spermiogenesis is conversion of spermatid into sperm. Spermatid into sperm or spermatozoa is called as spermiogenesis. What is spermiation? Spermiation is release of sperm from seminiferous tubule. That is called as spermiation, students. Beautiful concept. Yes or no, students? Beautiful concept. Yes or no? This much clarity you will get. Students, if you got the clarity, quickly smash the like button. And also, you're not putting comments. Also, put in the comments also. Okay, if you get a clarity, you can put diamond in the comment. Okay, that is called as spermatogenesis. Again, students, this is very easy to learn, very easy to question, very easy question. The real challenge is next one. All of you ready? Students, all of you ready for the next challenge? Students, all of you ready? See here again. Spermatogonia, primary spermatocyte, diploid, meiosis 1, second is spermatocyte, spermatids, mature sperm. Spermiogenesis happens here. This is what happens. This is how spermiogenesis. Okay, students, this is how spermiogenesis happen. Someone was asking me, right? Students, who was asking me? Vibs. This is, students, you want to learn? Again, I will not teach you in detail. I can show a little bit of how exactly happens. Again, no one will teach you this. No one will teach you this. Okay, students, can all of you see? This is called as your spermatid. This is spermatozoa, sperm. Can you see the conversion here? Students, can you see conversion here? Conversion happening. So what happens initially? Appearance of acrosome vessels and flagella in the plasmid, in the spermatid. There will be a flagella arising. On the top, we can see acrosome arising. Then growth of acrosome and flagella. Acrosomes had to grow later. Can you see acrosome is growing here? Flagella is growing right now. Then we have excess of cytoplasm. Remaining part is getting converted to flagella. And we can see the mitochondria development here. Mitochondria development in the middle. And eventually, we get the full mitochondria, part of middle piece. Acrosome here on the top of the head here. Head region with haploid nucleus. And mature sperm. Students, this is the actual spermiogenesis. Students, right? Students, I'm replying as much as possible. Okay, this right here is called as spermiogenesis. Students, spermiogenesis. Can you see the conversion of the spermatid, right? Conversion of spermatid into spermatozoa. Students, tell me yes or no. Classic, right? Classic. This is the entire information, students. We always go in detail. Students, is the voice proper? Students, sound check. Don't tell ghost and all. Students, clear to all of you? Yes or no? Nandu, all of you in the chat. Clear? Okay, students. Now, this is spermiogenesis. Can you see again here also? I've shown here also, students. Can you see? We have the spermatid, acrosome formation happens, tail formation happens, and we get the tail and acrosome here. Here also, students. See here also. Spermiogenesis, I've shown you here one more diagram. All of these diagrams are from your standard textbooks. You will not find them in NCERT, not find them anywhere. These are standard textbooks diagrams. So what if the tail is not formed? If the tail is not formed, the sperm are non-motile. They are waste. Sperm is waste that time. Okay. Can you see, students? See, this is your NCERT diagram. This is the standard textbook diagram. These are taken from your MBBS textbooks. These diagrams are taken from your MBBS textbooks also. MBBS textbooks. Okay. Now, students, all of you ready? Students, the next challenge. Students, all of you ready for the next challenge? Oogenesis. Again, students, if you understand oogenesis, you are sorted this chapter. This chapter is very easy if you understand oogenesis, students. Shall we start? All of you. Students, I forgot to get water today. Shall we start? Oogenesis. Students, first of all, tell me in the chat right now. How many of you feel oogenesis is difficult to understand? How many of you feel oogenesis is difficult to understand? Raise your hands in the chat. All of you. Oogenesis is the ghost, huh? Students, how many of you feel oogenesis is difficult? Energy in the mitochondria, because mitochondria is present in the middle piece. Middle piece has a mitochondria, right? Okay, students, let's just start. Students, first things first, I want all of you to understand when does the oogenesis start? What is the starting point of oogenesis? Oogenesis starts at the embryonic stage. When the females, all the beautiful females, all the beautiful females in the world, when all the beautiful females are at the embryo stage, tiny embryo, in that stage itself, the entire oogenesis starts. So it is a embryonic event. It is a embryonic event, students. Embryonic event. Okay. Now, oogenesis starts with again a germ cell, the female germ cell. The female germ cell is called as oogonia. Okay, students. Okay, oogenesis. Here we have the oogonia, students. Here we have the oogonia. Now, students, initially also, this oogonia undergoes mitosis to divide further. Students, I want all of you to write with me, students. I want all of you to write with me, right? Please write with me. Please write with me, okay? Okay. Initially, we have the mitosis here. Okay. So, we have the oogonia here. Second, diploid in nature. Mitosis, increase in number. Increase in number, students. Now, again, what happens? One such oogonia is differentiating. One such oogonia undergoes differentiation. It becomes bigger. Okay. Once such oogonia undergoes differentiation, it becomes bigger here. Can all of you students? Once such oogonia undergoes differentiation, it becomes bigger, diploid. Now this bigger one is called as primary oocyte. Primary oocyte, students. Primary oocyte. Now, students, in examination, if they tell you, right? If they tell you, students, in examination, what is the ploidy of primary oocyte? What will you tell, students? What will you tell? Primary oocyte is always diploid in nature, students. Primary oocyte is always diploid. Now, students, now this primary oocyte, now it is starting to undergo a process called as meiosis 1. Meiosis 1. Just starting, students. Why? Because this meiosis 1 actually, students, this meiosis 1 gets suspended. It gets suspended. It is like, stop it. You're not going further. It gets suspended at prophase 1. Students, it gets suspended at prophase 1. And in prophase 1, which actual stage, students? Under prophase 1, it gets suspended at diplotin stage. at diploiding students. So at diploiding stage, it gets arrested students. It gets arrested. Completely it is arrested. Okay. At diploiding stage, it is arrested. Now I want all of you to understand when all of this event is happening. This entire event, right? This entire event is happening at embryonic stage. Embryonic stage. Embryonic stage, students. Embryonic stage. Entire event. Okay. Now, students, now what happens? Now, this after this, there is childbirth, birth of the child here, birth of the female child, right? The female child will grow older. That is the childhood. Childhood happens. Okay, childhood happens. Childhood. Now, students, when the first particular, okay, the female, when it reaches the first time ever, the female reaches puberty, the first periods we say, right? The first periods ever, which happens around 13 to 12 years old. That time, the first puberty, first time the female reaches puberty, that time this meiosis 1 is finally completed. So meiosis 1 is stopped. It is suppressed until puberty. Until puberty, it is completely suppressed. It is suppressed, students. It is suppressed until puberty. Now, once the female child reaches puberty, what happens? This meiosis 1 is completed and it can give rise to two types of cells. It gives two types of cells at puberty. This female child at puberty, meiosis 1 is completed, two types of cells. One is called as secondary oocyte. Along with the secondary oocyte, there is the first polar body. And students, remember, this entire division is unequal. Second oocyte is large, whilst first polar body is very tiny, students. And it doesn't have any usage here. It doesn't have any usage at all. No usage at all, students. No usage at all. Okay. So we get secondary oocyte here. Now, students, all of you tell me in the chat right now. Okay. Now, students, what is the ploidy of secondary oocyte? All of you in the chat. See, I told you, primary oocyte is diploid. Meiosis is happening. Meiosis is happening, students. Meiosis is happening, right? At puberty, we get secondary oocyte. Now, what is the ploidy of secondary oocyte, students? The ploidy of secondary oocyte is always haploid in nature. So, it is going to be haploid in nature. Haploid means 23 chromosomes are there in that. Okay. Now, this till here, students, anyone, any doubt, students? Till here, anyone, any doubt? All of you. Students, till here, any doubt? Because I want all of you to be on the same page with me, understanding on the go. That is my promise to all of you. Okay. Students, till here, any doubt? Any one of you? If there is no doubt, shall we proceed ahead, students? Shall we proceed ahead? Okay. Now, this is happening at puberty. So, at puberty, secondary oocyte and first polar body. Now, one question arises, students. Where is it even happening? This is happening inside ovary. Students, this entire thing is happening inside the ovary. Okay, it is happening, students, right? It is happening inside the ovary. Very important, students, very important. Happening inside the ovary. Please remember that. Because primary oocyte is produced inside the ovary itself. Okay. First period is called as menarche. Okay, it's also happening during menarche. Menarche. Okay, menarche. Now, students, now what happens? Let's take the secondary oocyte right now. Secondary oocyte. Now, secondary oocyte, students, it will now start to undergo meiosis 2. Meiosis 2. Now, you will wonder, sir, why it is only starting, it is not getting over. It is not getting over, students. Again it's getting suspended. So that is why we say oogenesis is a disruptive cycle, disruptive process. It doesn't happen at one go. It becomes stop. It takes breaks, multiple breaks we need, no, multiple breaks. Okay. Now again, students, it is getting suspended at metaphase 2. Suspended at metaphase 2. Suspended at metaphase 2. Suspended at metaphase 2, students. Suspended at metaphase 2. Suspended. Now, when do you think the secondary oocyte is actually finishing the second meiosis? Second meiosis. When? If you read NCERT, students, if you read NCERT, it is not mentioned at all. It is never mentioned in NCERT in gametogenesis. In oogenesis, it is never mentioned. Never mentioned. Then where is it mentioned, students? Where is it mentioned? It is mentioned under the fertilization. Yes, it is mentioned. So students, understand what happens. This is the secondary oocyte. This is the secondary oocyte. When the secondary oocyte comes in contact with the sperm during fertilization, when secondary oocyte, students, when secondary oocyte comes in contact with the sperm during fertilization, that is when the meiosis 2 is completed. All of you, let's say if there is no fertilization at all. So students, tell me in the chat right now, if there is no fertilization, never, during periods, which type of oocyte is released? So if there is no fertilization at all, every single time in a female, the every monthly cycle, this secondary oocyte is released as such. This secondary oocyte is released as such itself in menstrual cycle. It is released. But if there is fertilization, this meiosis 2 is finally over, and this will give rise to two cells actually. This secondary oocyte will release, inside it will release the ovum, or we call it ooted is better word, ooted. So this is where you use ooted. Ooted or ovum word. You can never say ovum is released here. Ovum is released during fertilization. Never. Ovum is never released in the starting. It is always secondary oocyte which is released during the puberty or during the menstrual cycle. It is always secondary oocyte which is released. Always remember, students. Always remember. Okay. Now, students. Now, along with ovum or ooted, we also get the tiny unequal second polar body. We get the second polar body, students. Now a question to all of you, students. I told you the first polar body is produced in the ovary. Yes or no? Yes. Where do you think the second polar body is produced? Anyone who can tell me, students? I'll wait for 10 seconds. I'll wait for 20 seconds, but I want the answer. Students, where do you think second polar body is produced? Anyone? All of you. Students, use my notes, more than enough. Fallopian tube, exactly, students. This second polar body is produced in the oviduct or fallopian tube. Why? Because oviduct is a site for fertilization. Yes or no? So, in second polar body is produced in oviduct. Why? Because you remember I told you, in the ampulla region of oviduct or fallopian tube, in ampulla region, the fertilization happens. Yes or no? The sperm and the egg meet in fallopian tube. So I told you, This entire thing happens during fertilization. So all of you remember, students, all of you remember, the second polar body is produced in the fallopian tube or oviduct. Students, beautiful, right? This is how you understand, not by heart anything. This is how you understand the entire oogenesis. But students, this is incomplete right now. This is incomplete, all of you. This is incomplete right now, students, because along with oogenesis, there is one more process called as folliculogenesis. I'll teach you right now that one. Development of follicle. Students, any doubt till now? All of you. I don't think there should be any doubt here. More than this, this process cannot get simple than this. This is the most simplest way to understand the entire oogenesis. The most simplest way to understand the oogenesis. But it is incomplete still. It is incomplete. You will not be able to solve questions with the help of this. Now I will complete it, students. Now I'll complete it. Okay, oogenesis. Now, I want all of you to understand what is a follicle and what is a oocyte. Please understand, students, please understand. For that, I need a one second. Let me just get my dog's ball. Give me a minute. Hi, students. Okay, so students, I got my dog's ball. Okay, students, I got my dog's ball. So I think he wants to play now. Okay. Now, students, understand. Let's say this is a oocyte. This right here is a oocyte right now. Now, if I take a oocyte and I add a layer of granulosa layer on top of it, can you see, students? On top of the oocyte, if I add a granulosa layer, right? If I add a granulosa layer, now this becomes a follicle. Students, now this becomes a follicle. This is a follicle right now. This entire thing is called as a follicle. If I remove the outside granulosa layer, the inside part is called as a oocyte. The inside part is called as a oocyte. Okay. So I want all of you to understand, students. There is something called as primary follicle, secondary follicle, tertiary follicle, as lastly Graafian follicle. Graafian follicle. Okay. Now primary follicle has primary oocyte plus layer of granulosa cells. So primary follicle has primary oocyte inside and then layer of granulosa cells. Okay. Then what about secondary follicle, students? Secondary follicle has inside again primary oocyte plus more layer of granulosa cells. And students, here in secondary follicle, there's a theca formation also. Theca formation. Okay. Now in tertiary follicle, students want to understand. In tertiary follicle, what exactly happens? In tertiary follicle, what is happening is, in tertiary follicle, the primary oocyte is converted to secondary oocyte. Student, this part here. Remember, I told you primary oocyte is converted to secondary oocyte, right? Primary oocyte is converted to secondary oocyte. In between somewhere here, we get what? We get the tertiary follicle. This is where we get tertiary follicle here, students. Tertiary follicle right here, tertiary follicle. Then here we have Graafian follicle, obviously. Here we have Graafian follicle, obviously. Okay, here we have tertiary follicle right now here somewhere. Okay, so in tertiary follicle, what is have students? In tertiary follicle, right? In tertiary follicle, primary oocyte gets converted to secondary oocyte. Plus, so if I ask you right now, students, if I ask you right now, in tertiary follicle, let's say this is a oocyte, this secondary oocyte now. In the secondary oocyte, there are many layers now on top, many layers on top. This is a tertiary follicle. In tertiary follicle, can anyone tell me which oocyte do I have? Anyone? In tertiary follicle, which oocyte do I have, students? Which oocyte do I have? All of you. All of you students. In tertiary follicle, which oocyte do I have? In tertiary follicle, I have secondary oocyte. Please remember this concept. Okay. Please remember concept, student. Please remember this concept. Kushi, wait. Okay. In secondary tertiary follicle, we have secondary oocyte right now. Because primary is got converted to secondary right now. Now, students, here, the theca here, right? The theca becomes theca externa and theca interna. Basically, theca is dividing. Theca externa and theca interna, it is dividing. Theca is dividing here. Now, students, again in tertiary follicle also, we have something called as a fluid-filled cavity. Here we have a fluid-filled cavity called as antrum. So there is antrum for students. Okay, that is my dog, student. Sound is my dog outside. Okay, he's doing kui because I took his ball and I came. No, he wants his ball back. Okay, that is called as antrum. Fluid-filled cavity called as antrum is there here in tertiary follicle. Okay, now what about Graafian follicle, students? In the case of Graafian follicle, students, we have the secondary oocyte. Obviously, we have the secondary oocyte, students. Now, students, this secondary oocyte has a layer around it. It has a layer around it, and that layer is called as students, zona pellucida. So secondary oocyte has a layer called as zona pellucida, students. Okay, zona pellucida, students. Okay, is that clear to all of you? This is what happens: primary follicle, secondary follicle, tertiary follicle, Graafian follicle. Secondary oocyte, layer, zona pellucida. And students, this is what happens here. Can you see? Here follicle development is here. Follicle developments are happening here. This is your oocyte development. Here we can see follicle development. See, tertiary follicle development. Okay, if you can see here also. This is NCI diagram here. Now this is ovum has been changed to secondary oocyte. Okay, students, see here. Follicle developments. Can you see? Theca follicles here. Primary oocyte is present here. Zona pellucida layer is there. Then this primary oocyte gets converted to secondary oocyte here. Can you see? Secondary oocyte, fluid-filled cavity called as antrum. Yes or no, students? Fluid-filled cavity called as antrum. This is your tertiary follicle. Here. Yes or no? Then this outer part becomes Graafian follicle and it ruptures. Can you see it rupturing here? It ruptured. This is what we learn in the 14th day. Remember, students, in 14th day there is LH surge. LH surge happens. The ovulation happens. This is the ovulation. Okay. Graafian follicle ruptures and the secondary oocyte is released. Here. Now, what about the remaining part? This remaining part gets converted to corpus luteum. Like this, corpus luteum. Okay, converted to corpus luteum. Here is a side-by-side comparison. Oogonia, initially we have mitosis, primary oocyte is diploid, first meiotic arrested. It is arrested. Ovulation will not happen until puberty. At puberty, first polar body, secondary oocyte. At puberty, students, first polar body, secondary oocyte. Okay, then We have meiosis 2. Finally, meiosis 2 gets over during fertilization, and we get ovum and the second polar body, students. We get the second polar body, students. Here is a question to all of you, students. Before you solve the question, let me show you NCERT, students. My dog name, students, is Cash. My dog name is Cash, CASH. Students, is the board visible to all of you? So this is very easy, student. This is very easy to understand. See, gametogenesis. Students, I told you, all of you. Yes or no? Sure. The primary sex organ is testis. I told you always. Spermatogenesis begins at the puberty. The spermatogonia, male germ cell, spermatogonia present on the inside of the wall of seminiferous tubule, multiply by mitosis. See, starting we have mitosis, students. Starting there is mitosis, and increase in number. Each spermatogonia is diploid and contains 46. Some of the spermatogonia are called as primary spermatocyte, diploid again. Okay. Now they undergo meiosis 1. We get secondary spermatocyte. Okay. The second undergoes second mural division, meiosis 2. We get spermatid. Then spermatozoa, spermiogenesis. That's all. That is spermiation. Release of seminiferous tubule is called as spermiation. That is very easy, students. Now, students, I'll teach you oogenesis according to NCERT. According to NCERT, see the students. The process of the process of formation of mature female gamete is called as oogenesis. Okay. Oogenesis is initiated during embryonic stage, when couple of millions of gametes called as oogonia are formed within each fetal ovary. Okay. So oogonia are millions of oogonia are there. Okay, millions of oogonia are there. So no more oogonia are formed after birth. So after birth, oogonia are not added. Same number of oogonia is always there. Okay, same number of oogonia will always remain. Oogonia will never increase after that. Okay, now students, see this. These cells start division and enter into prophase 1. Told you, they will start meiosis 1, but they will stop at prophase 1, students. Okay. See here. See, of prophase 1 they'll stop here. Prophase 1 they'll stop your division and temporarily arrested at the stage called as primary oocyte is arrested here. Right. Now each primary oocyte then gets surrounded by a layer of granulosa cells and is called as primary follicle. Students, I've told you primary oocyte, right? If it gets a layer of granulosa cells, it is called as primary follicle. Same thing, students. A large number of these follicles degenerate. So primary follicle will start to degenerate throughout the years. So by the time the female child reaches the puberty, there are only at puberty 60,000 to 80,000 primary follicles are left. Okay, the primary follicle gets surrounded by more layer of granulosa cells, a new theca formation happens, and we get secondary follicle. Students, this is what I told here. This is what we learned here, students. Right? This is what we learned. More layer of granulosa cells, secondary oocyte, secondary follicle. The secondary follicle soon transforms into tertiary follicle. Students, very important line of very important line of NCERT. See this. The secondary follicle soon transforms into tertiary follicle, which is characterized by fluid-filled cavity called as antrum. The theca layer is organized into inner theca, interna, outer theca called as externa. Now, students, it is important to draw all of you your attention here. Students, all of your attention here, students. Students, all of you attention right here. See this. It is important to draw your attention that, see this, it is at this stage. What stage? Tertiary follicle stage, right? Tertiary follicle stage, because antrum and tertiary follicle, right? Tertiary follicle stage, that primary oocyte within the tertiary follicle, primary oocyte inside tertiary follicle grows in size and completes its first meiotic division. So students, the first meiotic division, the first meiosis is completed when, students? Tell me when. The first meiosis is completed in tertiary follicle. Very important of NCERT. Okay, it is an unequal division resulting in the formation of large haploid secondary oocyte. So we get secondary oocyte. Students, we get secondary oocyte in tertiary follicle and tiny first polar body. Okay, students, this is what I meant here. This is what I meant here, students. Here, can you see? Very important of NCERT. I told you here also, students, in tertiary follicle, the primary oocyte will finish the meiosis 1. It will finish the meiosis 1. It will finish the meiosis 1 and we get secondary oocyte. Students, so secondary oocyte is formed. Secondary oocyte is formed inside the tertiary follicle. Very beautiful line of NCERT. Many will miss it out, and if you don't understand that, you lose marks. So inside tertiary follicle, primary oocyte will finish the meiosis 1 and we get secondary oocyte. If you go back here, students, here meiosis 1 is completed, here we get secondary oocyte. This secondary oocyte in the starting, secondary oocyte is formed when? It is formed in tertiary follicle. Beautiful, students. Tell me, how beautiful is oogenesis, students? Tell me, how beautiful is oogenesis? Spermatogenesis is very simple. Very simple. But oogenesis is so beautiful. See here. We get secondary oocyte, in tertiary follicle in the starting. In the tertiary follicle. Later on it becomes Graafian follicle. Graafian follicle ruptures and ovulation happens. Eventually. So this is why I say read NCERT, students. This is why I say read NCERT always, you get better understanding here, serious. Okay. Its first meiotic division, unequal division, secondary oocyte in tiny first polar body. Now students, I want all of you to focus. Now fertilization part. See the students here. I want all of you to go here right now. Skip menstrual cycle for now. For now, let's skip menstrual cycle. Let's skip it. Now students, I want all of you to notice here. Okay. During copulation, coitus or the sex, semen is released in the penis into vagina, right? One second, students. Students, that line is very important. I want all of you to read that line. Students, see this. Read this line here, students. Okay. I'll just read from here. The process of fusion of sperm with the ovum is called as fertilization. During fertilization, a sperm comes in contact with the ZP, zona pellucida layer, okay, of the ovum and induces the change in the membrane that blocks the entry. This I'll teach you later on, students. Don't worry. The secretion of the acrosome helps in sperm entry into the cytoplasm of the ovum through the zona pellucida and the plasma membrane. So, students, basically, sperm is coming in contact. Sperm is coming in contact with the egg. Sperm is coming in contact with the secondary oocyte right now. Sperm is coming in contact with the secondary oocyte right now, students. Okay. Now read this line, students. This induces the completion of meiotic division. So during the fertilization, when the sperm is coming in contact with the secondary oocyte, see here, this induces the completion of meiotic division of the secondary oocyte. So secondary oocyte will finish the second meiosis. Okay. The second meiotic division is also unequal and results in the formation of second polar body and a haploid ovum or the ootid. Can you see, students, here? Or the ootid here, ootid, right? This is where it happens. This is where the second, student, this is where the second meiosis is getting over during fertilization. Just before fertilization, not during, before fertilization. Just before fertilization, when the sperm is coming in contact with the secondary oocyte, that is where it's getting over. Students, again see, very beautiful lines of NCERT. They should have actually written this in the previously, but they have not written it. What to do, students? Okay. No, coitus interest is different. Coitus interest next chapter. Quiz, yes, I will. Quiz, not sure here. Okay. Friends, I will do question back to students. Don't worry. Students, tell me your concept clarity. Tell me, students, how is your concept clarity right now? How is your concept clarity right now, students? So during fertilization, when second oocyte coming out of the sperm, this second meiosis is getting completed. We get ovum or ooted. This ooted and second polar body. This ooted nucleus of this ooted will fuse with the nucleus of the sperm and zygote is formed. That is called as syngamy. Syngamy happens later on. Okay, syngamy happens later on, students. Okay, students, here is a question. Okay, the division of primary oocyte result in, students, division of primary oocyte results in, when the primary oocyte divides, what do we get? We get unequal cells, one small or one large. Okay, do we get large haploid cell? Yes. Large haploid secondary oocyte and a small haploid polar body? Yes. Yes or no? So we get a large haploid secondary oocyte and a small haploid polar body we get. Okay. Now, how do ovulated oocytes get into uterus? How do ovulated oocytes get into uterus? I told you, students, yesterday's previous class, cilia movement. Cilia movement, students. With the help of ciliate movement, it can get in. Okay. Now, students, the primary oocyte completes its first meiotic division within. Students, tell me, when does the primary oocyte complete the first meiosis and become secondary oocyte? Which follicle? Students, which follicle? It happens in tertiary follicle. Do all of you remember? I told you, students, in tertiary follicle it gets completed, students. Here, again we go back here. In tertiary follicle, primary oocyte is converted to secondary oocyte. In tertiary follicle, students. In tertiary follicle, primary oocyte is converted to secondary oocyte. This question, same thing. The developing oocyte divides and releases the first polar body in. Students, the first polar body is released where? The first polar body is released in the tertiary follicle again. So do you realize it happens in tertiary follicle again? Because first polar body is produced along with the secondary oocyte. First polar body is produced along with secondary oocyte. It is happening in tertiary follicle again. It is happening in tertiary follicle again here. Secondary oocyte and first polar body in tertiary follicle again. So these are the concept questions. You can't by heart any of it. Okay. Oogenesis starts, students. Oogenesis starts at embryonic stage. Embryonic stage. Arrested stage called primary oocyte is formed. When do we get arrested stage, students? Arrested stage is present at the fetal growth again. Friends, I told you, arrested stage is at the fetal growth. Here you can see, students. Arrested stage happening at embryonic stage or fetal stage. Fetal stage. Fluid filled is a catastrophe, students. Where do you find fluid filled cavity? Fluid filled cavity is found in your tertiary follicle. Antrum. Antrum. Yes or no? Secondary oocyte is formed. Secondary oocyte is formed, students. When? Secondary oocyte is formed at puberty. Yes, secondary oocyte, sorry. That is secondary oocyte. Secondary oocyte is formed at puberty. Yes or no? See here. These are concept-based questions, high-level questions. Secondary oocyte is produced at puberty. Yes or no? At puberty. At puberty we get secondary oocyte inside tertiary follicle. Secondary oocyte inside tertiary follicle at puberty. This question. Oogenesis starts embryonic stage in the system. No? Okay. Ooted is formed along with. This is your homework question, students. This is your homework question for all of you. I want to see this answer in the comments, today's comments. This is the homework question, students. This is your homework question, all of you. Homework question. Ooted is formed along with. Okay. Now, before we go, one more question here, students. Very beautiful question. This is, what is released during ovulation? During ovulation, what do we get? Okay. Not today, no. Third part is that one. Today only this much. I don't want to confuse all of you. Okay. What is released during ovulation? During ovulation, students, we always get secondary oocyte. Yes or no? We get secondary oocyte. Secondary oocyte arrested at meiosis 1 or meiosis 2? Meiosis 2. Here. Arrested at meiosis 2, students. So at here. See, meiosis 1 is over. After meiosis 1, we get meiosis 2. Right? So meiosis 1 is over. That's how we get secondary oocyte. This secondary oocyte is released during ovulation, which is arrested at meiosis 2. Which is arrested at meiosis 2, students. That is my last question from my side. Students, these are called as beautiful questions. Okay. Now, students, before I go, students, I want all of you to tell me, students, right? There is a event coming up, students. All of you quickly smash the like button, first of all. All of you quickly smash the like button. I want to see 100 likes here. I want to see 100 likes here as soon as possible. Okay, students. Now, students, I'm teaching in DNA batch. DNA batch, I'll be teaching only botany. Don't worry. I will not teach zoology for all of you. Not bore all of you with zoology. In DNA batch, I'll be teaching only botany from my side. And if you're part of DNA batch, you already know my level of teaching there. In DNA batch, I go even more detailed. In DNA batch, I always teach so detailed. Every student is like, wow. Okay, especially botany. Tomorrow, I'll be teaching plant kingdom there, students. If you're not part of DNA batch, you can click here and join DNA batch as soon as possible here. See here. Click here and you can join DNA batch here. The price right now is 4,000. Again, it will go back to 7,000 very soon. 7,000. So 4,000 is like very minimum amount. Here you can get unlimited questions. Unlimited questions. All the notes. Tattva book PDF copy. Pedagogy. More than 500 ebooks you'll get. All in a test series you'll get. Remember, students, if you want to buy test series later on during your NEET preparation, test series itself will cost you how much? 2,000 rupees. So for now, 4,000 rupees you're getting an entire batch itself. Okay, so all of you should know. And students, if you go here on the channel, crazy thing is happening here, students. If you go here and if you check, we are giving all the we are doing a celebration called as Vedantu, right? Celebration here. I'll be anchoring here, students. I'll be here. You can see my face and all the channels there. Okay, so I'll be there in this event. I'll be anchoring a lot of things there. So quickly, students, all of you quickly smash the like button here and wait for this session. Very beautiful session this will happen. We will facilitate all the NEET toppers, JE toppers, Olympiad toppers, everyone here. Okay, students, that's it for my class today, students. That's it from my side today, right? I will meet all of you in the next class. Until then, students, all of you take care. And this is what I want in comments. I want all of you to tell me in the chat how was the class in the comment section. Okay, students. So bye bye, all of you. Take care. I hope concept was clear to all of you. This is my way of teaching. If you love it, you will love it. Okay. So in part 3, I will do fertilization in detail. I will teach you fertilization beyond NCERT in the next class. So until then, students, take care, all of you. Bye bye, and have a great day ahead, all of you.